Well, hello, everybody. Welcome back to our ongoing look at the chapters for this semester. And this time we are looking at chapter 25, a discussion of the history of life on Earth, as the chapter title suggests. In this chapter, we're going to look at some ideas that have been proposed as to how life may have originated, how abiotic or non-living chemistry could give rise to biotic or organic chemistry. Then we'll look at the fossil record, and we'll look at that very briefly because you have already looked at the nature of the fossil record through the paleontology lab, which we've already completed. So we don't really need to discuss that in great detail, but then we'll get into a look at some of the high points along the way that we can identify from the fossil record and from our understanding of how life has evolved over time in that history. And then finally, we'll take a look at the genetic basis for the evolution of life, which is something that we will scratch the surface of in this chapter, but in later chapters, particularly towards the end of the semester, we'll dig a little bit deeper into the role of genes in guiding or as part of the evolution of life. I want to avoid using the term guiding because there is no real guidance here. It's one of the issues that people have with evolution, the fact that people want certainty and they want to see guidance and they want to see purpose, and evolution has none. And that's one of the things that we will touch on at the end of this chapter when we talk about the nature of evolution and whether there is a direction to evolution, whether there is a purpose in terms of the changes that occur in organisms, or whether there's something else that we need to be aware of. Anyway, let's get started. So one of the important things that we need to recognize is that the only certainty as far as life and our planet is concerned is that it's going to constantly change. That is one of the things that we have to bear in mind. And as a consequence of changes in the Earth, life has to respond to that. Because if it doesn't respond to it, the organisms that are extant at that point will become extinct because they are unable to accommodate the changes. The changes push them to extinction. What is also interesting to recognize, however, is not only does geological changes in the Earth drive the evolution of organisms, organisms themselves at various points along the way in the history of life have led to changes in the planet that have in turn driven changes in the direction of evolution, have changed the trajectory of evolution. And we'll touch on some of those points. So the interaction between organisms and the planet is basically a reciprocal one, where largely the geological changes, the environmental changes that occur on the planet drive changes in the trajectory of evolution, but likewise, organisms have at various points in time generated environmental changes that have in turn changed the trajectory of evolution. So those are the two things that we need to be very aware of when we think in terms of what is or what are the driving forces behind the change in organisms, what we would call natural selection and evolution over time. So one of the first things that we really need to discuss when we start thinking about the origin of life on the planet is the fact that all of the organic chemistry that we see now, and in fact, the origin of organic chemistry as we know it today, originated with looking at the chemistry of life. And a philosophy arose as people started to examine the chemistry of organisms back in the particularly 1600s and 1700s and on into the 1800s. And that philosophy was called vitalism. And it basically said that organic chemistry was only possible with organisms, that you could not have organic chemistry, you could not have organic molecules in the absence of organisms. And that life itself arose spontaneously from pre-existing life. Now, there was no indication as to where that pre-existing life came from. Just that, for instance, if you left a piece of meat out for a while, at some point in time, you would start to observe maggots emerging from the meat. And the argument here was that maggots were spontaneously generated from the meat. In other words, life is spontaneously generated from pre-existing life in the form of the meat that you left out. There was no connection between the presence of the maggots and the fact that flies had been visiting the meat for the whole of that time and potentially may have had some role in the appearance of the maggots. And the fact that the maggots developed into flies was observed, but the connection to the origin of maggots was completely lost. And so this idea was very, very prominent right up until the mid-1800s. Around 1843, 1848 is the date that I recall, running on memory here. And what really started to discredit this whole idea of vitalism was the simple observation that you could generate organic molecules by mixing inorganic molecules together. And the first example of this occurred quite by accident, as many times major discoveries actually do occur. And this was the work of a German chemist by the name of Friedrich Wöhler, who generated the organic substance urea accidentally by leaving two chemicals sitting in a beaker and forgetting to wash the beaker out, and coming back later and realizing that what he had in that beaker were crystals of urea that had apparently spontaneously appeared in the beaker. Now, the reason why this was significant to Wöhler was that he was actually interested in urea because he was working on the chemical substances that you could identify in urine. And he used animal urine. I'm sure he made his own contributions to his research as well. But he would take urine and allow the water to evaporate and then take a look at what was left behind. And one of the substances that he identified was urea. And so he was very, very familiar with crystals of urea. So when he discovered urea in this beaker, where he was absolutely sure he had not had any urine previously, number one, it was very surprising, but then number two, he became curious as to how that might have happened. And so he repeated deliberately the mixing of the chemicals together, and sure enough, he generated urea. And then he worked on trying to improve his ability to make urea, to increase the yield of urea. And we still talk about the Wöhler synthesis as a way of making urea, because urea is a very common component of industrial fertilizers because it's a really good source of nitrogen that is accessible to plants. But the whole point here is that Wöhler had generated an organic molecule in the absence of life, something that should not have been possible under the vitalism idea. And so once it was recognized that you could actually generate organic molecules by mixing inorganic molecules together, people started playing around with trying to generate other types of organic molecules, developing organic syntheses. And over time, particularly in Germany, where the chemists became exceptionally good at doing this, organic chemistry started to emerge as a new branch of chemistry. And in fact, the vast majority of our industrial chemistry today is, in fact, a form of organic chemistry. And so it is the basis for a lot of the chemical industries that we are dependent upon. But it begins with Wöhler's simple observation that you could make urea by mixing two inorganic chemicals together. Now, what's the big deal with that? Why is that so important to us as biologists? Well, ultimately, it was accepted once Darwin published his ideas on natural selection and evolution, along with Wallace. The thought then started to play in the minds of biologists and chemists that if it is possible to create organic molecules in the absence of life by mixing inorganic chemicals together, and life over time has evolved from simple to complex, at some point you've got to go back to the point where life originates, where you see the first signs of cellular life. And that point, if you go any further backwards, there is an absence of cellular life, and all you have is inorganic chemistry. And so in 1921, two individuals, Oprin and Haldane, Oprin was Russian, Haldane was English, proposed a hypothesis that said that the chemical conditions of the primordial Earth, which was thought to be millions of years old at that point, the final age of the Earth would not be really figured out until the 1950s and 1960s, and even then, a very accurate age of the Earth was really not developed until just recently, within the last 10 or 15 years. But the whole idea here for Haldane and Oprin was that the chemical conditions that existed on that primordial Earth when it first condensed out of the leftovers from the formation of the sun and other parts of the solar system, those chemical conditions must at some point have been suitable for the generation of the precursor molecules that would ultimately come together, aggregate together to give rise to the first primitive life forms. And that it must be possible to demonstrate somehow that chemistry could in fact take place. Now, of course, you can't go back in time and take a look at the situation on the primordial Earth, but it ought to be possible, if we have an understanding of what the primordial life might have been like, to set up an experiment that would replicate those conditions in a laboratory setting, and then try to establish whether those conditions could in fact give rise to organic molecules, precursor molecules for life. The individual who demonstrated that this was possible was a man named Stanley Miller. He did this work as his graduate work, working on his PhD. And his supervising professor, Harold Urey, and he at the University of Chicago developed a experimental apparatus that allowed them to set up a sample atmosphere similar to what was considered to be at the time the atmosphere of the primitive Earth, and then allow that system to run for a period of time, usually for about a week or so, and periodically tap into whatever was being produced in order to test it chemically to see what was being produced, if anything was produced. This is what the device looked like. Haldane and Oprin proposed that water would be present, because we all know that water is fundamental to life. It is essential, and water has to be present, preferably in its liquid form, but at least water has to be present. Now, the primordial Earth was considered to be very hot, and almost certainly was very hot because it was cooling down from condensation and also from being hit by large bodies, which would create very hot conditions. So any water that existed, if it existed for a period of time as liquid, would almost certainly be turned back into water vapor. And so in the Miller-Urey apparatus, water is present as a vapor. Then in the experimental reaction chamber, which is this chamber up in the top right-hand corner of the device, they also included gases, methane, ammonia, and hydrogen, because these were the gases that were thought to have been the components of the primordial atmosphere. They also included an electrical discharge, because if you've ever seen pictures of volcanic eruptions or videos of volcanic eruptions, you'll notice that there are large static electricity discharges, lightning bolts, coming out of the stream of volcanic ash that's being ejected from the volcano into the atmosphere. It creates static electricity in this column of ash, and it gets released as electrical discharges from lightning bolts. And so, given that the primordial Earth was very hot and almost certainly extremely volcanic in activity, there would be a lot of electrical discharges in that primitive atmosphere. So they included the electrical discharge. You can see that then whatever is produced in the reaction chamber is collected by condensing the water vapor and whatever dissolves in it, returning back to the vessel containing the water, so that the water could be returned back into the reaction vessel with steam. But of course, the products of the chemical reactions would then start to accumulate in the beaker where the water is being boiled. And you could also tap into the cooled water to find out whatever substance is being produced. And if this device was allowed to run for 7 days or thereabouts, you would notice that the solution would change from a clear water-based solution to something that was initially a little bit yellow, more orange, and then a darker, almost brown color, and it would get thicker, more viscous as the time progressed. And so clearly something was being made. And when they tapped into the solution, what they found was that the products of the reactions going on in the reaction vessel were things like amino acids, hydrocarbons, simple hydrocarbons. And over time, Miller would continue with this work once he got a faculty position, and he would start playing with different mixes of gases as people's understanding of the atmosphere of the primordial Earth started to change and started to include other gases like carbon monoxide and carbon dioxide, along with methane, which we know are ejected from volcanoes, some water, of course, some sulfur dioxides, which are also present in the gases from volcanoes. And when you mix those together under these conditions, you actually increase the range of organic molecules that you can make. You can make simple sugars. Miller even identified molecules similar to the nitrogenous bases that are found in the nucleotides of DNA and RNA. So the range of organic molecules that can be produced is actually quite broad under these conditions. And Miller would continue with this work for the whole of his life. And he was quite convinced that this was the best explanation for the chemical origin of life, that the early precursors were the product of these sorts of chemical reactions occurring either in the primordial atmosphere of the Earth or in small pockets of water on the surface of the Earth. And when Miller died, he actually sent boxes of samples that he had not yet analyzed from his experiments to some of his former graduate students and asked them to continue examining this, because he was convinced that the range of organic molecules that could be produced in this fashion would eventually look very similar to that would be necessary for generating organic chemistry, life chemistry, from inorganic chemistry. So this is a confirmation, at least, of one of the 4 steps that are necessary to go from abiotic conditions, absence of life, to conditions that are supportive of life, to the appearance of life itself. Now, I skipped over the slide where we identify those 4 conditions. So let me go back and take a look at these. So here's the 4 circumstances that have to take place in sequence. So first, you have to have the ability to synthesize organic molecules in the absence of life. Waller suggested that that was possible. Subsequent research also said that that was most definitely possible. Miller now has demonstrated with his device that under the conditions that was suspected to occur on the primordial Earth, that should have been possible, at least when replicated in the laboratory. The next thing is then to polymerize these small precursor molecules into larger polymers, the macromolecules that we recognize are the major organic components of cells. So proteins, nucleic acids, and carbohydrates. Those are the 3 main polymers that are found in cells. Of course, we also have the hydrocarbons in the form of lipids, and it would be nice to be able to synthesize those as well. Miller has already suggested in his device that that's possible. We don't need those to be polymerized, but at least having the presence of hydrocarbons and particularly lipids would be a good thing as well. Then eventually these macromolecules have to come together in aggregates that show similarities to cells. because at some point, these molecules must have condensed into aggregates that started to take on the behaviors that we associate with cells until we cross the line from non-living aggregates to early cells. Where that line exists, we don't know, although there are people looking for the position of that line. But in laboratory experiments, at least, we ought to be able to demonstrate that aggregates can form that show at least some of the behaviors that we associate with cells. And then ultimately, because we have molecules like DNA and RNA that are capable of replicating themselves, we need to have the origin of self-replication. And that needs to be in the form initially of molecular self-replication, but ultimately replication of aggregates as a consequence of the nature of the molecules present in them, including molecules that are capable of self-replication. If all of those events occur in sequence, and obviously they must have occurred in sequence here on Earth because life exists, then you can at least establish a series of hypotheses that lead from abiotic chemistry through simple organic chemistry to more complicated organic chemistry in the form of these aggregates to self-replicating molecules and the emergence of cells. So we've already looked at Miller's work. Let's take a look at the possibility of polymerization. Well, before we get to polymerization, perhaps I really, and I've got this in the notes in this order, I really ought to touch on some other ideas outside of Miller's work, because remember, Miller's proposing that this is the synthesis of these small molecular molecules or is taking place either in the atmosphere or it's taking place in small volumes of water that are being heated on the surface of the Earth. One of the things that we've now discovered by looking at the age of rocks and the type of rocks that are present on the surface of the Earth is that while we don't have any rocks that date all the way back to the origin of the Earth around 4.58 billion years ago, which is the currently accepted age of the Earth, we do have rocks that are only a few 100 million years removed from the origin of the Earth. So the oldest rocks that have been dated date to around 4.2 billion years in age. And some of those rocks can be found along the coast of Maine in a couple of very small regions along the coast of Maine. There are some rocks that date to around that time. And what's interesting about those rocks is that some of the minerals that are present in those rocks could only have formed if the rocks themselves formed underneath a substantial layer of water. Not just a few inches of water, but feet of water, perhaps even hundreds of feet of water. And what these rocks are suggesting is that large bodies of water may have actually been stable on the surface of the Earth far earlier than we imagined or thought possible. That in fact, while it is true that the Earth was really hot and a lot of the water that arrived on Earth would be vaporized and be part of the atmosphere, there may have been regions where substantial bodies of water may have been able to aggregate fairly early in the history of life. Now, why that's important is because about 30 years ago, we discovered a whole new ecosystem on the floor of the ocean below 12,000 feet. And why that's important is because up until this point in time, it was always thought that all life on Earth was dependent on solar energy, captured by plants through photosynthesis and then distributed in various ways, depending on your feeding habits and what material you fed on to gain carbon and energy. And these ecosystems in these depths below 12,000 feet could not be dependent on anything that involved solar energy. They were just too deep. Light doesn't penetrate that far. Light penetrates about 500 feet, maybe 700 feet. into the ocean. Below that, certainly below 1,000 feet, you have to take your own light source because there is no light available. So if you get to 12,000 feet or more, you can forget there being light energy. Now, one other source of energy might have been what's called marine snow. These are remnants of organisms that over time settle down through the ocean to the ocean floor. But again, this would be a very limiting source of energy, and these ecosystems contained fish, crabs, very large worms known as bloodworms, bacteria and archaea. There's a broad array of organisms, and it would be very, very difficult to imagine that these ecosystems could be supported by the limited amount of energy obtained from the marine snow. What it turns out is that these ecosystems are dependent on chemicals dissolved in hot water that is coming out from the ocean floor in what are called hydrothermal vents, or to use the more common name, black smokers. The reason why they're called that is because the water is black in color because of all the dissolved minerals. Now, some of the chemicals, particularly H2S, hydrogen sulfide, or rotten egg gas, can be used as a source of energy. There are bacteria that extract electrons out of H2S in the same way that plants extract electrons out of H2O and use those electrons to reduce carbon dioxide to organic molecules, simple sugars, from which they can create all the other organic molecules that they need. And there are a group of bacteria and archaea that can do exactly the same thing. And the archaea that live around these thermal vents are those types of archaea, and they form the basis of these ecosystems because they either become food for some of the animals that live around there, or they live as symbionts in the bodies of these large tube worms, these bloodworms that are bright red because they have hemoglobin in them, and provide a source of energy for the worms, which then become the basis for ecosystems as a food source for the other animals. And we now know that these hydrothermal vents are widely distributed around those regions of the ocean floor where there are tectonic plates separating from each other. So the classic example here is the Mid-Atlantic Ridge that runs down the center of the Atlantic Ocean. There, two plates are separating from each other, the North American plate and the European plate, and lava is extruded through the gap as the plates move away from each other. But water also percolates down into cracks that form in the ocean floor as a consequence of this movement. As it percolates down through these cracks, of course, it gets heated by the underlying molten rock. And as the water heats up, it dissolves minerals in the rock because hot water can dissolve the minerals. And then those minerals, that water then gets shot up back through other cracks. And as it hits the near freezing water at that depth, the minerals condense out and form these tubes that then become the origin of the black smokers. As the minerals condense out and form the tubes, the water keeps venting out of the tubes. The archaea take advantage of the minerals as the water cools down to around 140 degrees Celsius. They are then able to survive in the water and tap into the H2S as a source of energy. And they become then the basis of the ecosystems that form around these vents. So here is another source of heat-driven chemistry. And people have proposed, and this hypothesis is gaining more traction, that this may actually be the origin of organic life. And there's a really good reason, well, two really good reasons for considering this as a potential source of the abiotic chemistry that would give rise to organic life. Number one, we know that there's a source of heat, there's a source of simple chemicals. We could mix those chemicals together because it's the same chemicals that ultimately Miller would be mixing in his reaction vessel. The sulfates, the carbon dioxide, the carbon monoxide, the volcanic gases, they're all present in this water as well. And so if they could form the precursor molecules for biotic life in Miller's apparatus, they could certainly form the precursor molecules in a similar sort of setup under a large body of water, so long as there is a heat source. And of course, there's obviously a heat source here. The added advantage of doing this in a body of water is that that water acts as a protective agent, particularly from ultraviolet radiation. And it is thought that the young sun was an intense source of ultraviolet radiation, certainly more intense than it is currently. And ultraviolet radiation would tend to break down and destroy these simple molecules, and also break down any polymers that might form. So this is an alternative hypothesis to the Miller-Urey hypothesis. And it is a hypothesis that, as I said, is gaining strength and gaining greater recognition as people continue to consider all the possibilities of where these organic molecules may have come from that gave rise to life. The final hypothesis, and this one also has a number of supporters, and there is evidence to back this up, is that those organic molecules may have simply come from remnants of the early solar system as the solar system started to condense out. What is interesting is that we can find clouds of organic molecules scattered throughout the solar system and generally throughout the universe. And you can see the chemical signature of them as you look around in space. And we know that some of these organic molecules can also arrive on Earth as a consequence of collisions with comets and collisions with meteorites. There are two basic types of meteorites. There are iron meteorites, really metallic, really heavy, but then there are carbonaceous meteorites as well. And in those carbonaceous meteorites, you find amino acids and other molecules that are considered to be precursors of life. There's the simple molecules that would give rise to the larger polymers. So it is not impossible to imagine that some of the molecules, if not all of the molecules, that seeded the Earth and created the possibility for organic life did not originate here, but originated in space and arrived in the form of comets and meteorites. So that's another hypothesis. But the one that has the greatest acceptance right now is the hydrothermal vent hypothesis of the three. That's the one that seems to have the greatest support at this point. So once we have these simple molecules, the next step, of course, is to polymerize them. And the polymerization actually takes place using a mechanism not dissimilar from what we use in our industrial chemistry, using metal ions. So one of the way in which we generate organic molecules, and particularly polymers, through our industrial chemistry is to use metal ions like zinc, iron itself, nickel, copper, cobalt, to act as catalysts for the polymerization, the binding together of the monomers to form polymers. Well, it turns out that those ions are very common in clays. You find aluminum, you find iron, you find magnesium, metals that we already know, or metal ions that we already know can act as catalysts for organic chemistry because they are the basis of our industrial chemistry. In some cases, they function at normal, quote unquote, normal room temperature. In other cases, they need a source of heat, but that would not be a problem on the primordial Earth because heat is obviously available. All that is necessary is to have concentrations of the monomers of sufficient quantity to allow for polymerization. And again, if you've got things like the accumulations by the Miller process or accumulations around hydrothermal vents, you've got your source of heat. If you've got in the case of the Miller model, if you've got these condensing out and collecting in small pools of water on the surface of the earth where it might be stable. Then, of course, they could come in contact with the metal ions in the clays and the rock that these pools of water are sitting in. So the idea of polymerizing the monomers into polymers and larger macromolecules is not difficult to explain. It really is the basis of our industrial chemistry. So if we can do it in our industrial chemistry with these metal ions, then the presence of the metal ions in the clays, in the rocks, and the conditions that are proposed either by Miller's hypothesis or the hydrothermal vent hypothesis, and even by the extra Earth, origin from outside of Earth hypothesis. Still, the presence of the metal ions as potential catalysts allows us to propose a hypothesis for where the polymerization may have taken place and how the early polymers may have come together. The next thing then is to have these polymers aggregate together to form bodies that resemble cells. In the lab, when we create these, we refer to them as protobions. So proto, primitive or precursor, bions refers to life. So these are primitive or precursor aggregations that are not living, but have characteristics that we associate with cells. So the simplest one is simply to mix lipids together, particularly phospholipids. If you just mix simple lipids together, they form liposomes, which have a single layer of lipids as a membrane bounding a lipid droplet in which things can be dissolved. If you use phospholipids, of course, phospholipids will organize themselves into the lipid bilayer that we associate with all cell membranes, and you can create liposomes in that fashion. If you add a catalyst, and one of the catalysts that's been used in laboratory situations is actually a clay that is derived from breaking down volcanic material, then you actually can increase the formation of the liposomes, or vesicles as they're referred to here. But the term vesicle is a more generalized term because in this case, the vesicles could be formed from lipids, or they could be formed from proteins, because proteins will form a vesicle structure as well. But if you add these clay particles, and here's the diagram that illustrates that. Here is the formation of vesicles without the clay. Here's the formation of vesicles with the clay. And you can see that it is much easier for the vesicles to form in the presence of the clay particles acting as a catalyst, a catalytic surface for the vesicles to form on. And of course, based on our previous argument, the fact that ions in the clays can act as catalysts for the polymerization of the polymers, then the clays themselves now are acting as catalysts for those polymers to then condense into protobions. If we have the formation of protobions, what we can also see is that you can have simple biochemistry associated with this. You can also have aggregates associated with these, or you can form aggregates that associate with other organic molecules. So this diagram shows the incorporation of an RNA molecule. That's the orange looking material. It's been stained so that it can be identified, being captured inside one of these protobions. And you can see the quote unquote membrane that outlines the protobiont, and then the DNA inside. And then there are other lipid particles captured inside the vesicle as well. One of the other things that you also see with spontaneously forming vesicles is that they do reproduce themselves. It turns out that as they accumulate lipids into their vesicle membrane, there is a limit to how big they can get. And once they get to a certain size, they will automatically bud off smaller vesicles. This is a consequence of the physics and the dynamics of the formation of the vesicles. And so here you can see a vesicle giving birth, if you want to use that term, to smaller vesicles. as it accumulates lipids out of the solution that it's forming in. In some cases, these lipid layers can even form semi-permeable layers and become discriminating in terms of what they allow in and what they don't allow in. And if they form in solutions that contain simple precursors to biochemical pathways, and one of the simplest ones that's been demonstrated is the polymerization of glucose to form starch. If in your vesicles you incorporate the enzyme that's responsible for polymerizing glucose into starch, these selective vesicles can bring in the glucose, and then the enzyme can form starch. If you then add in a hydrolase that breaks the starch down, the starch that's formed inside can then be broken down and, under the right conditions, be exported back out of the vesicle again. In other words, these are examples of very similar biochemistry that's being carried out by a vesicle that clearly is not a cell, but is showing some features that we would ascribe to cells. So over time, seeing as how we can demonstrate the reality of this in the laboratory, if enough time exists, and of course we now recognize that time is not an issue because of the age of the Earth, then there would be enough time for these sorts of spontaneous chemical processes to take place, and eventually aggregates to start to form that would ultimately give rise to cells. The final step is self-replicating molecules. Now, we today think of the genetic material of cells as being DNA, with the exception of some viruses that use RNA as their genetic material. But in General Biology 1, you hopefully were introduced to the concept of ribozymes. These are RNA molecules that are catalytic, and they are involved in processes like assembling amino acids into proteins. It's the little RNA molecules in the ribosomes that are actually acting as the enzymes, in the cutting and splicing of exons, so cutting the transcript in eukaryotes to cut out the introns and splicing the exons together by little complexes called spliceosomes that contain RNA molecules. There it's the RNA molecules that are acting as enzymes. These are all examples of known ribozymes in cells. What is interesting is that RNA not only can function as an enzyme, it is also much more heat stable. And given that the early Earth was very hot, it would be more likely that RNA of the two nucleic acids would be present than DNA. DNA is only heat stable until around 90 degrees Celsius. At that temperature, all the hydrogen bonds that cross-link the two polynucleotide strands via the nucleotides break down, and the two nucleotide strands fall apart, and you no longer have functional DNA molecules. And so if you want to think in terms of the earliest molecules of hereditary, the earliest genetic material, you think RNA rather than DNA. And of course, RNA, we now know, can function as an enzyme. And it turns out that some of these RNA molecules are self-replicating. And this has actually been examined in the laboratory, where sequences of known ribozymes that have an autocatalytic function, meaning they can act on themselves to self-replicate, have been used as the basis for generating some artificial RNA molecules that also have self-replicating properties. These are then placed in test tubes along with the 4 RNA nucleotides, and the molecules are allowed to then self-replicate. These are then examined to find out which of the synthetic molecules have the best enzyme activity. Those are then used as the basis for generating a new generation of self-replicating RNA molecules, which are then allowed to self-replicate. Those are then examined to find out which ones are the best at self-replicating. And over the course of these experiments, the researchers were able, through this process of artificial selection, to generate RNA molecules that had a 10-fold increase in ability to self-replicate by simply, for each generation of self-replicating synthetic RNA molecules, selecting those that showed the greatest ability to self-replicate and using those as the basis for the next generation, a form of artificial selection. Now, why that's important is that it indicates that self-replicating molecules may actually be subject to natural selection, that you may actually be able to have natural selection in the absence of cellular life. And RNA molecule is a really good candidate for this, because what you see here in this diagram is an RNA molecule. And the first thing that you should recognize is that the RNA molecule has a three-dimensional shape. And that three-dimensional shape is a consequence of base pairing between complementary RNA nucleotides in certain regions of the RNA molecule. So technically speaking, this RNA molecule has both a genotype, a sequence of nucleotides, which is what the genotype is, and a phenotype, the physical shape of the molecule as a consequence of that genotype, that sequence of nucleotides, which allows for the base pairing between different regions of the RNA molecule. So it has a genotype and a phenotype. And of course, if you have genotypes and phenotypes, then you have the possibility of natural selection, because different nucleotide sequences will give rise to different phenotypes, which will in turn affect its function and its ability to self-replicate. And that was demonstrated in the laboratory experiment through artificial selection. So certainly we can propose this as a basis for natural selection of self-replicating molecules. After all, it was artificial selection, artificial breeding that both Darwin and Wallace were familiar with, more so Darwin than Wallace, that formed the basis for the subsequent development of the concept of natural selection as an extension of what life can do, what the environment can do, that man can do much faster by artificially selecting organisms for breeding. The environment can do the same thing. It just takes more time and is not as purposeful as what would be the case with what man does. We deliberately identify the individuals that we want to breed. In the experiment, they deliberately identified the RNA molecules that they wanted to use for further examination. But in natural selection, it's whatever happens to be favored by the environment. And that would be true for natural selection amongst RNA molecules. So an RNA world, which is likely the beginnings of genetics, does not actually need cellular life, although ultimately, of course, genetic material would be encapsulated into membrane-bound vesicles and would subsequently give rise to cells, because there would be a distinct advantage both to the RNA molecules and to the aggregates to work together rather than to try and exist separately. So all of these steps are simple hypotheses that we can at least examine in the laboratory to determine whether it is possible under laboratory conditions that are similar to those that we think existed in the primordial Earth. But they are only hypotheses. This is not and should not be construed as a listing of what actually happened. It's what we can do in the laboratory. But certainly steps like these must have happened. Why? Because life exists. And in fact, given what we've been able to demonstrate through laboratory experiments, and given what we've been able to observe on the one planet where we know there's life, our own, biologists and chemists that are interested in the question of the origin of life basically think along this line. If chemical conditions on a planet or a well, let's just say a planet, are conducive to the chemical synthesis of life, it is almost inevitable. The numbers that I've seen are anywhere from 70% to 99.9% inevitable that life will emerge. And this is the reason why astrobiologists, biologists that are interested in looking for life off our planet, are very interested in Mars and also two moons, one around Saturn and one around Jupiter, that have, at least in the past, in the case of Mars and in the case of these two moons, presently have conditions that may have been conducive to the evolution of life. And obviously people want to go and take a look. And NASA is in fact, obviously currently exploring that possibility on Mars, and is also looking at launching a mission to take a look at least at Enceladus, that's the moon around Saturn, that has apparently conditions that may be supportive of life. Obviously, a lot more interesting research to be done in that area. So these are the basic ideas that we have about how abiotic chemistry may have given rise to organic chemistry. That it did take place is absolutely certain, because life exists on our planet. These are our currently accepted hypotheses that we think provide an explanation, at least to some degree, as to how it might have taken place. So let's stop for our first break. And here is the concept check question. Remember, at each break, there will be a concept check question that I'll pose. And then after the break, I'll rejoin you with the answer to the concept check question before we move on to the next part. So here's the concept check question for this break. What hypothesis did Miller test in his classic experiment? It's a really good question. Let me stop here, and I'll see you on the other side of the break with the answer.